Section 0 of Gray's Anatomy, Part 1. Anatomy of the Human Body, Part 1, by Henry Gray. Preface to the 20th edition. Since the publication of the first English edition of this work in 1858, and the first American edition in 1859, great advances in the subject of anatomy have been made, especially in microscopic anatomy and the anatomy of the embryo. This knowledge was embodied from time to time in the successive editions until finally considerable portions of the text, sometimes sections, were devoted to these subjects. However, the main text has always remained primarily a descriptive anatomy of the human body. In the present edition, the special sections on embryology and histology have been distributed among the subjects under which they naturally belong. New matter on physiological anatomy, laws of bone architecture, the mechanics and variations of muscles have been added, occupying much of the space formerly devoted to the sections on applied anatomy. The sections on the ductless glands and the nervous system have been largely rewritten. In the latter, a more rational presentation of the sympathetic nervous system has been achieved through the use of diagrams and descriptions based on physiological and pharmacological work. The central connections of the spinal and cranial nerves are also emphasized. Illustrations have been added wherever important points could be made more clear, and throughout the work colored pictures have been even more extensively used than heretofore. In this respect, special mention might be made of the central nervous system and the section on the muscles. In the section on syndesmology, 6 illustrations are used from Quain's Anatomy through the courtesy of the publishers, Messrs. Longmans, Green & Company of London. The use of the B.N.A. nomenclature in English has been retained practically unchanged in this edition, and important references to the literature have been added at the end of each section. As a practical work on the subject for the student, Gray's Anatomy has always been recognized and appreciated. The plan originally formulated, which has proved so successful, has been adhered to as much as possible. It is interesting to note that although Henry Gray saw only the first edition, much of the original text persists, and many of his illustrations are still in use. Bearing this in mind, it has been the endeavor of the editor to supply only such changes as advances in the science made necessary in order that this work may reflect the latest accessions to anatomical knowledge. W. H. L., Baltimore, 1918. Introduction. The term human anatomy comprises a consideration of the various structures which make up the human organism. In a restricted sense, it deals merely with the parts which form the fully developed individual and which can be rendered evident to the naked eye by various methods of dissection. Regarded from such a standpoint, it may be studied by 2 methods. 1. The various structures may be separately considered, systematic anatomy. Or, 2. The organs and tissues may be studied in relation to one another, topographical or regional anatomy. It is, however, of much advantage to add to the facts ascertained by naked eye dissection those obtained by the use of the microscope. This introduces 2 fields of investigation, that is, the study of the minute structure of the various component parts of the body, histology, and the study of the human organism in its immature condition, that is, the various stages of its intrauterine development from the fertilized ovum up to the period when it assumes an independent existence, embryology. Owing to the difficulty of obtaining material illustrating all the stages of this early development, gaps must be filled up by observations on the development of lower forms, comparative embryology, or by consideration of adult forms in the line of human ancestry, comparative anatomy. The direct application of the facts of human anatomy to the various pathological conditions which may occur constitutes the subject of applied anatomy. Finally, the appreciation of structures on or immediately underlying the surface of the body is frequently made the subject of special study, surface anatomy. Systematic anatomy. The various systems of which the human body is composed are grouped under the following headings. Osteology, the bony system or skeleton. Syndesmology, the articulations or joints. Myology, the muscles. With the description of the muscles, it is convenient to include that of the fascia which are so intimately connected with them. Angiology, the vascular system, comprising the heart, blood vessels, lymphatic vessels, and lymph glands. Neurology, the nervous system, The organs of sense may be included in this system. Splanknology, the visceral system. Topographically, the viscera form two groups, that is, the thoracic viscera and the abdomino-pelvic viscera. The heart, a thoracic viscus, is best considered with the vascular system. The rest of the viscera may be grouped according to their functions: A, the respiratory apparatus; B, the digestive apparatus; and C, the urogenital apparatus. Strictly speaking, the third subgroup should include only such components of the urogenital apparatus as are included within the abdomino-pelvic cavity, but it is convenient to study, under this heading, certain parts which lie in relation to the surface of the body, that is, the testes and the external organs of generation. For descriptive purposes, the body is supposed to be in the erect posture, with the arms hanging by the sides and the palms of the hands directed forward. The median plane is a vertical antero-posterior plane passing through the center of the trunk. This plane will pass approximately through the sagittal suture of the skull, and hence any plane parallel to it is termed a sagittal plane. A vertical plane at right angles to the median plane passes, roughly speaking, through the central part of the coronal suture or through a line parallel to it. Such a plane is known as a frontal plane, or sometimes as a coronal plane. A plane at right angles to both the median and frontal planes is termed a transverse plane. The terms anterior or ventral and posterior or dorsal are employed to indicate the relation of parts to the front or back of the body or limbs, and the terms superior or cephalic and inferior or caudal to indicate the relative levels of different structures. Structures nearer to or farther from the median plane are referred to as medial or lateral, respectively. The terms superficial and deep are strictly confined to descriptions of the relative depth from the surface of the various structures. External and internal are reserved almost entirely for describing the walls of cavities or of hollow viscera. In the case of the limbs, the words proximal and distal refer to the relative distance from the attached end of the limb. End of section 0.
Section 1 of Gray's Anatomy, Part 1. Anatomy of the Human Body, Part 1, by Henry Gray. Section 1. Embryology. The term embryology in its widest sense is applied to the various changes which take place during the growth of an animal from the egg to the adult condition. It is, however, usually restricted to the phenomena which occur before birth. Embryology may be studied from two aspects. One, that of ontogeny, which deals only with the development of the individual, and two, that of phylogeny, which concerns itself with the evolutionary history of the animal kingdom. In vertebrate animals, the development of a new being can only take place when a female germ cell or ovum has been fertilized by a male germ cell or spermatozoon. The ovum is a nucleated cell, and all the complicated changes by which the various tissues and organs of the body are formed from it after it has been fertilized are the result of two general processes, viz., segmentation and differentiation of cells. Thus, the fertilized ovum undergoes repeated segmentation into a number of cells which at first closely resemble one another, but are, sooner or later, differentiated into two groups: one, somatic cells, the function of which is to build up the various tissues of the body, and two, germinal cells, which become embedded in the sexual glands, the ovaries in the female and the testes in the male, and are destined for the perpetuation of the species. Having regard to the main purpose of this work, it is impossible in the space available in this section to describe fully or illustrate adequately all the phenomena which occur in the different stages of the development of the human body. Only the principal facts are given, and the student is referred for further details to one or other of the textbooks on human embryology. 1. The animal cell. All the tissues and organs of the body originate from a microscopic structure (brackets, the fertilized ovum), close brackets, which consists of a soft jelly-like material enclosed in a membrane and containing a vesicle or small spherical body, inside which are one or more denser spots. This may be regarded as a complete cell. All the solid tissues consist largely of cells essentially similar to it in nature, but differing in external form. In the higher organisms, a cell may be defined as a nucleated mass of protoplasm of microscopic size. Its two essentials, therefore, are a soft jelly-like material similar to that found in the ovum, and usually styled cytoplasm, and a small spherical body embedded in it, and termed a nucleus. Some of the unicellular protozoa contain no nuclei, but granular particles which, like true nuclei, stain with basic dyes. The other constituents of the ovum, viz., its limiting membrane and the denser spot contained in the nucleus, called the nucleolus, are not essential to the type cell, and in fact many cells exist without them. Cytoplasm (protoplasm) is a material probably of variable constitution during life, but yielding on its disintegration bodies chiefly of proteid nature. Lecithin and cholesterin are constantly found in it, as well as inorganic salts, chief among which are the phosphates and chlorides of potassium, sodium, and calcium. It is of a semi-fluid, viscid consistence, and probably colloidal in nature. The living cytoplasm appears to consist of a homogeneous and structureless ground substance, in which are embedded granules of various types. The mitochondria are the most constant type of granule, and vary in form from granules to rods and threads. Their function is unknown. Some of the granules are proteid in nature and probably essential constituents. Others are fat, glycogen, or pigment granules, and are regarded as adventitious material taken in from without, and hence are styled cell inclusions or paraplasm. When, however, cells have been fixed by reagents, a fibrillar or granular appearance can often be made out under a high power of the microscope. The fibrils are usually arranged in a network or reticulum, to which the term spongioplasm is applied, the clear substance in the meshes being termed hyaloplasm. The size and shape of the meshes of the spongioplasm vary in different cells, and in different parts of the same cell. The relative amounts of spongioplasm and hyaloplasm also vary in different cells, the latter preponderating in the young cell, and the former increasing at the expense of the hyaloplasm as the cell grows. Such appearances in fixed cells are no indication whatsoever of the existence of similar structures in the living, although there must have been something in the living cell to give rise to the fixed structures. The peripheral layer of a cell is in all cases modified either by the formation of a definite cell membrane, as in the ovum, or more frequently in the case of animal cells by a transformation, probably chemical in nature, which is only recognizable by the fact that the surface of the cell behaves as a semi-permeable membrane. Nucleus. The nucleus is a minute body embedded in the protoplasm, and usually of a spherical or oval form, its size having little relation to that of the cell. It is surrounded by a well-defined wall, the nuclear membrane. This encloses the nuclear substance (nuclear matrix), which is composed of a homogeneous material in which is usually embedded one or two nucleoli. In fixed cells, the nucleus seems to consist of a clear substance or karyoplasm, and a network or karyomytome. The former is probably of the same nature as the hyaloplasm of the cell, but the latter, which forms also the wall of the nucleus, differs from the spongioplasm of the cell substance. It consists of fibres or filaments arranged in a reticular manner. These filaments are composed of a homogeneous material known as linin, which stains with acid dyes, and contains embedded in its substance particles which have a strong affinity for basic dyes. These basophil granules have been named chromatin or basichromatin, and owe their staining properties to the presence of nucleic acid. Within the nuclear matrix are one or more highly refracting bodies termed nucleoli, connected with the nuclear membrane by the nuclear filaments. They are regarded as being of two kinds. Some are mere local condensations (net knots) of the chromatin. These are irregular in shape and are termed pseudonucleoli. Others are distinct bodies differing from the pseudonucleoli both in nature and chemical composition. They may be termed true nucleoli and are usually found in resting cells. The true nucleoli are oxyphil, i.e. they stain with acid dyes. Most living cells contain, in addition to their protoplasm and nucleus, a small particle which usually lies near the nucleus and is termed the centrosome. In the middle of the centrosome is a minute body called the centriole, and surrounding this is a clear spherical mass known as the centrosphere. The protoplasm surrounding the centrosphere is frequently arranged in radiating fibrillar rows of granules, forming what is termed the attraction sphere. Reproduction of cells. Reproduction of cells is effected either by direct or by indirect division. In reproduction by direct division, the nucleus becomes constricted in its centre, assuming an hourglass shape, and then divides into two. This is followed by a cleavage or division of the whole protoplasmic mass of the cell, and thus two daughter cells are formed, each containing a nucleus. These daughter cells are at first smaller than the original mother cell, but they grow and the process may be repeated in them, so that multiplication may take place rapidly. Indirect division, or karyokinesis (karyomitosis), has been observed in all the tissues: generative cells, epithelial tissue, connective tissue, muscular tissue, and nerve tissue. It is possible that cell division may always take place by the indirect method. The process of indirect cell division is characterized by a series of complex changes in the nucleus, leading to its subdivision. This is followed by cleavage of the cell protoplasm. Starting with the nucleus in the quiescent or resting stage, these changes may be briefly grouped under the four following phases. 1. Prophase. The nuclear network of chromatin filaments assumes the form of a twisted skein or spirum, while the nuclear membrane and nucleolus disappear. The convoluted skein of chromatin divides into a definite number of V-shaped segments or chromosomes. The number of chromosomes varies in different animals, but is constant for all the cells in an animal of any given species. In man, the number is given by Fleming and Duesberg as 24. Coincidentally with or preceding these changes, the centriole, which usually lies by the side of the nucleus, undergoes subdivision, and the two resulting centrioles, each surrounded by a centrosphere, are seen to be connected by a spindle of delicate achromatic fibres, the achromatic spindle. The centrioles move away from each other, one toward either extremity of the nucleus, and the fibrils of the achromatic spindle are correspondingly lengthened. A line encircling the spindle midway between its extremities or poles is named the equator, and around this the V-shaped chromosomes arrange themselves in the form of a star, thus constituting the mother star or monaster. 2. Metaphase. Each V-shaped chromosome now undergoes longitudinal cleavage into two equal parts or daughter chromosomes, the cleavage commencing at the apex of the V and extending along its divergent limbs. 3. Anaphase. The daughter chromosomes, thus separated, travel in opposite directions along the fibrils of the achromatic spindle toward the centrioles, around which they group themselves, and thus two star-like figures are formed, one at either pole of the achromatic spindle. This constitutes the diaster. The daughter chromosomes now arrange themselves into a skein or spirum, and eventually form the network of chromatin which is characteristic of the resting nucleus. 4. Telophase. The cell protoplasm begins to appear constricted around the equator of the achromatic spindle, where double rows of granules are also sometimes seen. The constriction deepens, and the original cell gradually becomes divided into two new cells, each with its own nucleus and centrosome, which assume the ordinary positions occupied by such structures in the resting stage. The nuclear membrane and nucleolus are also differentiated during this phase. End of section 1. Recording by Ruth Golding. Section 2 of Gray's Anatomy, Part 1. Anatomy of the Human Body, Part 1, by Henry Gray. Embryology. 2. The ovum. The ova are developed from the primitive germ cells which are embedded in the substance of the ovaries. Each primitive germ cell gives rise, by repeated divisions, to a number of smaller cells termed oogonia, from which the ova or primary oocytes are developed. Human ova are extremely minute, measuring about 0.2 millimeters in diameter, and are enclosed within the egg follicles of the ovaries. As a rule, each follicle contains a single ovum, but sometimes two or more are present. Footnote: see description of the ovary on a future page. End of footnote. By the enlargement and subsequent rupture of a follicle at the surface of the ovary, an ovum is liberated and conveyed by the uterine tube to the cavity of the uterus. Unless it be fertilized, it undergoes no further development and is discharged from the uterus. But if fertilization take place, it is retained within the uterus and is developed into a new being. In appearance and structure, the ovum differs little from an ordinary cell, but distinctive names have been applied to its several parts. Thus, the cell substance is known as the yolk or ooplasm, the nucleus as the germinal vesicle, and the nucleolus as the germinal spot. The ovum is enclosed within a thick, transparent envelope, the zona striata or zona pellucida, adhering to the outer surface of which are several layers of cells derived from those of the follicle, and collectively constituting the corona radiata. Yolk. The yolk comprises 1. The cytoplasm of the ordinary animal cell with its spongioplasm and hyaloplasm. This is frequently termed the formative yolk. 2. The nutritive yolk or deutoplasm, which consists of numerous rounded granules of fatty and albuminoid substances embedded in the cytoplasm. In the mammalian ovum, the nutritive yolk is extremely small in amount, and is of service in nourishing the embryo in the early stages of its development only, whereas in the egg of the bird there is sufficient to supply the chick with nutriment throughout the whole period of incubation. The nutritive yolk not only varies in amount, but in its mode of distribution within the egg. Thus, in some animals it is almost uniformly distributed throughout the cytoplasm. In some it is centrally placed and is surrounded by the cytoplasm. In others it is accumulated at the lower pole of the ovum, while the cytoplasm occupies the upper pole. A centrosome and centriole are present and lie in the immediate neighbourhood of the nucleus. Germinal vesicle. The germinal vesicle, or nucleus, is a large spherical body which at first occupies a nearly central position, but becomes eccentric as the growth of the ovum proceeds. Its structure is that of an ordinary cell nucleus, viz. It consists of a reticulum or karyomitome, the meshes of which are filled with karyoplasm, while connected with, or embedded in, the reticulum are a number of chromatin masses or chromosomes, which may present the appearance of a skein, or may assume the form of rods or loops. The nucleus is enclosed by a delicate nuclear membrane, and contains in its interior a well-defined nucleolus or germinal spot. Coverings of the ovum. The zona striata or zona pellucida is a thick membrane which, under the higher powers of the microscope, is seen to be radially striated. It persists for some time after fertilization has occurred, and may serve for protection during the earlier stages of segmentation. It is not yet determined whether the zona striata is a product of the cytoplasm of the ovum, or of the cells of the corona radiata, or both. The corona radiata consists of 2 or 3 strata of cells. They are derived from the cells of the follicle, and adhere to the outer surface of the zona striata when the ovum is set free from the follicle. The cells are radially arranged around the zona, those of the innermost layer being columnar in shape. The cells of the corona radiata soon disappear. In some animals they secrete, or are replaced by, a layer of adhesive protein, which may assist in protecting and nourishing the ovum. The phenomena attending the discharge of the ova from the follicles belong more to the ordinary functions of the ovary than to the general subject of embryology, and are therefore described with the anatomy of the ovaries. Footnote: see description of the ovary on a future page. End of footnote. Maturation of the ovum. Before an ovum can be fertilized, it must undergo a process of maturation or ripening. This takes place previous to or immediately after its escape from the follicle, and consists essentially of an unequal subdivision of the ovum, first into 2 and then into 4 cells. 3 of the 4 cells are small, incapable of further development, and are termed polar bodies or polar-cytes, while the 4th is large and constitutes the mature ovum. The process of maturation has not been observed in the human ovum, but has been carefully studied in the ova of some of the lower animals, to which the following description applies. It was pointed out on page 37 that the number of chromosomes found in the nucleus is constant for all the cells in an animal of any given species, and that in man the number is probably 24. This applies not only to the somatic cells, but to the primitive ova and their descendants. For the purpose of illustrating the process of maturation, a species may be taken in which the number of nuclear chromosomes is 4. If an ovum from such be observed at the beginning of the maturation process, it will be seen that the number of its chromosomes is apparently reduced to 2. In reality, however, the number is doubled, since each chromosome consists of 4 granules grouped to form a tetrad. During the metaphase, see page 37, each tetrad divides into 2 dyads, which are equally distributed between the nuclei of the 2 cells formed by the first division of the ovum. One of the cells is almost as large as the original ovum, and is named the secondary oocyte. The other is small, and is termed the first polar body. The secondary oocyte now undergoes subdivision, during which each dyad divides and contributes a single chromosome to the nucleus of each of the 2 resulting cells. This second division is also unequal, producing a large cell which constitutes the mature ovum, and a small cell, the second polar body. The first polar body frequently divides while the second is being formed, and as a final result 4 cells are produced, viz. The mature ovum and 3 polar bodies, each of which contains 2 chromosomes, i.e. 1/2 the number present in the nuclei of the somatic cells of members of the same species. The nucleus of the mature ovum is termed the female pronucleus. End of section 2. Recording by Ruth Golding. Section 3 of Gray's Anatomy, Part 1. Anatomy of the Human Body, Part 1, by Henry Gray. Embryology. 3. The spermatozoan. The spermatozoa or male germ cells are developed in the testes and are present in enormous numbers in the seminal fluid. Each consists of a small but greatly modified cell. The human spermatozoan possesses a head, a neck, a connecting piece or body, and a tail. The head is oval or elliptical but flattened, so that when viewed in profile it is pear-shaped. Its anterior 2/3 are covered by a layer of modified protoplasm, which is named the head cap. This in some animals, e.g. the salamander, is prolonged into a barbed spear-like process or perforator, which probably facilitates the entrance of the spermatozoan into the ovum. The posterior part of the head exhibits an affinity for certain reagents and presents a transversely striated appearance, being crossed by 3 or 4 dark bands. In some animals, a central rod-like filament extends forward for about 2/3 of the length of the head, while in others a rounded body is seen near its centre. The head contains a mass of chromatin, and is generally regarded as the nucleus of the cell surrounded by a thin envelope. The neck is less constricted in the human spermatozoan than in those of some of the lower animals. The anterior centriole, represented by 2 or 3 rounded particles, is situated at the junction of the head and neck, and behind it is a band of homogeneous substance. The connecting piece or body is rod-like and is limited behind by a terminal disc. The posterior centriole is placed at the junction of the body and neck, and, like the anterior, consists of 2 or 3 rounded particles. From this centriole an axial filament, surrounded by a sheath, runs backward through the body and tail. In the body, the sheath of the axial filament is encircled by a spiral thread, around which is an envelope containing mitochondria granules and termed the mitochondria sheath. The tail is of great length, and consists of the axial thread or filament surrounded by its sheath, which may contain a spiral thread or may present a striated appearance. The terminal portion or end piece of the tail consists of the axial filament only. Krause gives the length of the human spermatozoon as between 52 μ and 62 μ, the head measuring 4 to 5 μ, the connecting piece 6 μ, and the tail from 41 μ to 52 μ. By virtue of their tails, which act as propellers, the spermatozoa are capable of free movement, and if placed in favourable surroundings, e.g. in the female passages, will retain their vitality and power of fertilizing for several days. In certain animals, e.g. bats, it has been proved that spermatozoa retained in the female passages for several months are capable of fertilizing. The spermatozoa are developed from the primitive germ cells which have become embedded in the testes, and the stages of their development are very similar to those of the maturation of the ovum. The primary germ cells undergo division and produce a number of cells termed spermatogonia, and from these the primary spermatocytes are derived. Each primary spermatocyte divides into two secondary spermatocytes, and each secondary spermatocyte into two spermatids or young spermatozoa. From this it will be seen that a primary spermatocyte gives rise to 4 spermatozoa. On comparing this process with that of the maturation of the ovum, it will be observed that the primary spermatocyte gives rise to 2 cells, the secondary spermatocytes, and the primary oocyte to 2 cells, the secondary oocyte and the first polar body. Again, the 2 secondary spermatocytes, by their subdivision, give origin to 4 spermatozoa, and the secondary oocyte and first polar body to 4 cells, the mature ovum and 3 polar bodies. In the development of the spermatozoa, as in the maturation of the ovum, there is a reduction of the nuclear chromosomes to 1/2 of those present in the primary spermatocyte. But here the similarity ends, for it must be noted that the 4 spermatozoa are of equal size, and each is capable of fertilizing a mature ovum, whereas the 3 polar bodies are not only very much smaller than the mature ovum, but are incapable of further development, and may be regarded as abortive ova. 4. Fertilization of the ovum. Fertilization consists in the union of the spermatozoon with the mature ovum. Nothing is known regarding the fertilization of the human ovum, but the various stages of the process have been studied in other mammals, and from the knowledge so obtained, it is believed that fertilization of the human ovum takes place in the lateral or ampullary part of the uterine tube, and the ovum is then conveyed along the tube to the cavity of the uterus, a journey probably occupying 7 or 8 days, and during which the ovum loses its corona radiata and zona striata, and undergoes segmentation. Sometimes the fertilized ovum is arrested in the uterine tube and there undergoes development, giving rise to a tubal pregnancy. Or it may fall into the abdominal cavity and produce an abdominal pregnancy. Occasionally the ovum is not expelled from the follicle when the latter ruptures, but is fertilized within the follicle and produces what is known as an ovarian pregnancy. Under normal conditions, only one spermatozoon enters the yolk and takes part in the process of fertilization. At the point where the spermatozoon is about to pierce, the yolk is drawn out into a conical elevation termed the cone of attraction. As soon as the spermatozoon has entered the yolk, the peripheral portion of the latter is transformed into a membrane, the vitelline membrane, which prevents the passage of additional spermatozoa. Occasionally a second spermatozoon may enter the yolk. thus giving rise to a condition of polyspermy. When this occurs, the ovum usually develops in an abnormal manner and gives rise to a monstrosity. Having pierced the yolk, the spermatozoon loses its tail, while its head and connecting piece assume the form of a nucleus containing a cluster of chromosomes. This constitutes the male pronucleus, and associated with it there are a centriole and centrosome. The male pronucleus passes more deeply into the yolk, and coincidentally with this, the granules of the cytoplasm surrounding it become radially arranged. The male and female pronuclei migrate toward each other, and, meeting near the centre of the yolk, fuse to form a new nucleus, the segmentation nucleus, which therefore contains both male and female nuclear substance. The former transmits the individualities of the male ancestors, the latter those of the female ancestors, to the future embryo. By the union of the male and female pronuclei, the number of chromosomes is restored to that which is present in the nuclei of the somatic cells. End of section 3. Recording by Ruth Golding. Section 4 of Gray's Anatomy, Part 1. Anatomy of the Human Body, Part 1, by Henry Gray. Embryology. 5. Segmentation of the fertilized ovum. The early segmentation of the human ovum has not yet been observed, but judging from what is known to occur in other mammals, it may be regarded as certain that the process starts immediately after the ovum has been fertilized, i.e., while the ovum is in the uterine tube. The segmentation nucleus exhibits the usual mitotic changes, and these are succeeded by a division of the ovum into two cells of nearly equal size. Footnote: in the mammalian ova, the nutritive yolk or deutoplasm is small in amount and uniformly distributed throughout the cytoplasm. Such ova undergo complete division during the process of segmentation and are therefore termed holoblastic. In the ova of birds, reptiles and fishes, where the nutritive yolk forms by far the larger portion of the egg, the cleavage is limited to the formative yolk and is therefore only partial. Such ova are termed meroblastic. Again, it has been observed in some of the lower animals that the pronuclei do not fuse, but merely lie in apposition. At the commencement of the segmentation process, the chromosomes of the two pronuclei group themselves around the equator of the nuclear spindle and then divide. An equal number of male and female chromosomes travel to the opposite poles of the spindle, and thus the male and female pronuclei contribute equal shares of chromatin to the nuclei of the two cells which result from the subdivision of the fertilized ovum. End of footnote. The process is repeated again and again, so that the two cells are succeeded by 4, 8, 16, 32, and so on, with the result that a mass of cells is found within the zona striata, and to this mass the term morula is applied. The segmentation of the mammalian ovum may not take place in the regular sequence of 2, 4, 8, etc., since one of the two first formed cells may subdivide more rapidly than the other, giving rise to a 3 or a 5 cell stage. The cells of the morula are at first closely aggregated, but soon they become arranged into an outer or peripheral layer, the trophoblast, which does not contribute to the formation of the embryo proper, and an inner cell mass from which the embryo is developed. Fluid collects between the trophoblast and the greater part of the inner cell mass, and thus the morula is converted into a vesicle, the blastodermic vesicle. The inner cell mass remains in contact, however, with the trophoblast at one pole of the ovum. This is named the embryonic pole, since it indicates the situation where the future embryo will be developed. The cells of the trophoblast become differentiated into two strata: an outer, termed the syncytium or syncytiotrophoblast, so named because it consists of a layer of protoplasm studded with nuclei but showing no evidence of subdivision into cells, and an inner layer, the cytotrophoblast or layer of Langhans, in which the cell outlines are defined. As already stated, the cells of the trophoblast do not contribute to the formation of the embryo proper. They form the ectoderm of the chorion and play an important part in the development of the placenta. On the deep surface of the inner cell mass, a layer of flattened cells, the entoderm, is differentiated and quickly assumes the form of a small sac, the yolk sac. Spaces appear between the remaining cells of the mass, and by the enlargement and coalescence of these spaces a cavity, termed the amniotic cavity, is gradually developed. The floor of this cavity is formed by the embryonic disc, composed of a layer of prismatic cells, the embryonic ectoderm, derived from the inner cell mass and lying in apposition with the entoderm. The primitive streak, formation of the mesoderm. The embryonic disc becomes oval and then pear-shaped, the wider end being directed forward. Near the narrow posterior end, an opaque streak, the primitive streak, makes its appearance, and extends along the middle of the disc for about 1/2 of its length. At the anterior end of the streak there is a knob-like thickening, termed Henson's knot. A shallow groove, the primitive groove, appears on the surface of the streak, and the anterior end of this groove communicates, by means of an aperture, the blastophore, with the yolk sac. The primitive streak is produced by a thickening of the axial part of the ectoderm, the cells of which multiply, grow downward, and blend with those of the subjacent entoderm. From the sides of the primitive streak a third layer of cells, the mesoderm, extends lateralward between the ectoderm and entoderm. The caudal end of the primitive streak forms the cloacal membrane. The extension of the mesoderm takes place throughout the whole of the embryonic and extra-embryonic areas of the ovum, except in certain regions. One of these is seen immediately in front of the neural tube. Here the mesoderm extends forward in the form of two crescentic masses, which meet in the middle line so as to enclose behind them an area which is devoid of mesoderm. Over this area the ectoderm and entoderm come into direct contact with each other and constitute a thin membrane, the buccopharyngeal membrane, which forms a septum between the primitive mouth and pharynx. In front of the buccopharyngeal area, where the lateral crescents of mesoderm fuse in the middle line, the pericardium is afterward developed, and this region is therefore designated the pericardial area. A second region where the mesoderm is absent, at least for a time, is that immediately in front of the pericardial area. This is termed the proamniotic area, and is the region where the proamnion is developed. In man, however, a proamnion is apparently never formed. A third region is at the hind end of the embryo, where the ectoderm and entoderm come into apposition and form the cloacal membrane. The blastoderm now consists of 3 layers, named from without inward: ectoderm, mesoderm, and entoderm. Each has distinctive characteristics and gives rise to certain tissues of the body. Footnote: the mode of formation of the germ layers in the human ovum has not yet been observed. In the youngest known human ovum, viz. That described by Bryce and Teacher, all 3 layers are already present, and the mesoderm is split into its 2 layers. The extra-embryonic coelom is of considerable size, and scattered mesodermal strands are seen stretching between the mesoderm of the yolk sac and that of the chorion. End of footnote. Ectoderm. The ectoderm consists of columnar cells which are, however, somewhat flattened or cubical toward the margin of the embryonic disc. It forms the whole of the nervous system, the epidermis of the skin, the lining cells of the sebaceous, sudoriferous and mammary glands, the hairs and nails, the epithelium of the nose and adjacent air sinuses, and that of the cheeks and roof of the mouth. From it also are derived the enamel of the teeth and the anterior lobe of the hypophysis cerebri, the epithelium of the cornea, conjunctiva and lacrimal glands, and the neuroepithelium of the sense organs. Entoderm. The entoderm consists at first of flattened cells which subsequently become columnar. It forms the epithelial lining of the whole of the digestive tube, excepting part of the mouth and pharynx and the terminal part of the rectum, which are lined by involutions of the ectoderm, the lining cells of all the glands which open into the digestive tube, including those of the liver and pancreas, the epithelium of the auditory tube and tympanic cavity, of the trachea, bronchi, and air cells of the lungs, of the urinary bladder and part of the urethra, and that which lines the follicles of the thyroid glands and thymus. Mesoderm. The mesoderm consists of loosely arranged branched cells surrounded by a considerable amount of intercellular fluid. From it the remaining tissues of the body are developed. The endothelial lining of the heart and blood vessels and the blood corpuscles are, however, regarded by some as being of entodermal origin. As the mesoderm develops between the ectoderm and entoderm, it is separated into lateral halves by the neural tube and notochord, presently to be described. A longitudinal groove appears on the dorsal surface of either half, and divides it into a medial column, the paraxial mesoderm, lying on the side of the neural tube, and a lateral portion, the lateral mesoderm. The mesoderm in the floor of the groove connects the paraxial with the lateral mesoderm, and is known as the intermediate cell mass. In it the genito-urinary organs are developed. The lateral mesoderm splits into two layers, an outer or somatic, which becomes applied to the inner surface of the ectoderm, and with it forms the somatopleure, and an inner or splanchnic, which adheres to the entoderm, and with it forms the splanchnoplure. The space between the two layers of the lateral mesoderm is termed the coelom. End of section 4. Recording by Ruth Golding.
Section 5 of Gray's Anatomy, Part 1. Anatomy of the Human Body, Part 1, by Henry Gray. Part 6. The Neural Groove and Tube. In front of the primitive streak, two longitudinal ridges, caused by a folding up of the ectoderm, make their appearance, one on either side of the middle line. These are named the neural folds. They commence some little distance behind the anterior end of the embryonic disc, where they are continuous with each other, and from there gradually extend backward, one on either side of the anterior end of the primitive streak. Between these folds is a shallow median groove, the neural groove. The groove gradually deepens as the neural folds become elevated, and ultimately the folds meet and coalesce in the middle line and convert the groove into a closed tube, the neural tube or canal, the ectodermal wall of which forms the rudiment of the nervous system. After the coalescence of the neural folds over the anterior end of the primitive streak, the blastopore no longer opens on the surface, but into the closed canal of the neural tube, and thus a transitory communication, the neuronteric canal, is established between the neural tube and the primitive digestive tube. The coalescence of the neural folds occurs first in the region of the hind brain, and from there extends forward and backward. Toward the end of the third week, the front opening, anterior neuropore, of the tube finally closes at the anterior end of the future brain, and forms a recess which is in contact for a time with the overlying ectoderm. The hinder part of the neural groove presents for a time a rhomboidal shape, and to this expanded portion the term sinus rhomboidalis has been applied. Before the neural groove is closed, a ridge of ectodermal cells appears along the prominent margin of each neural fold. This is termed the neural crest or ganglion ridge, and from it the spinal and cranial nerve ganglia and the ganglia of the sympathetic nervous system are developed. By the upward growth of the mesoderm, the neural tube is ultimately separated from the overlying ectoderm. The cephalic end of the neural groove exhibits several dilatations, which, when the tube is closed, assume the form of three vesicles. These constitute the three primary cerebral vesicles, and correspond respectively to the future forebrain, prosencephalon, midbrain, mesencephalon, and hindbrain, rhombencephalon. The walls of the vesicles are developed into the nervous tissue and neuroglia of the brain, and their cavities are modified to form its ventricles. The remainder of the tube forms the medulla spinalis, or spinal cord. From its ectodermal wall, the nervous and neuroglial elements of the medulla spinalis are developed while the cavity persists as the central canal. Part 7. The notochord. The notochord consists of a rod of cells situated on the ventral aspect of the neural tube. It constitutes the foundation of the axial skeleton, since around it the segments of the vertebral column are formed. Its appearance synchronizes with that of the neural tube. On the ventral aspect of the neural groove an axial thickening of the entoderm takes place. This thickening assumes the appearance of a furrow, the chordal furrow, the margins of which come into contact and so convert it into a solid rod of cells, the notochord, which is then separated from the entoderm. It extends throughout the entire length of the future vertebral column, and reaches as far as the anterior end of the midbrain, where it ends in a hook-like extremity in the region of the future dorsum celli of the sphenoid bone. It lies at first between the neural tube and the entoderm of the yolk sac, but soon becomes separated from them by the mesoderm, which grows medialward and surrounds it. From the mesoderm, surrounding the neural tube and notochord, the skull and vertebral column, and the membranes of the brain and medulla spinalis are developed. Part 8. The primitive segments. Toward the end of the second week, transverse segmentation of the paraxial mesoderm begins, and it is converted into a series of well-defined, more or less cubical masses, the primitive segments, which occupy the entire length of the trunk on either side of the middle line from the occipital region of the head. Each segment contains a central cavity, myocele, which, however, is soon filled with angular and spindle-shaped cells. The primitive segments lie immediately under the ectoderm on the lateral aspect of the neural tube and notochord, and are connected to the lateral mesoderm by the intermediate cell mass. Those of the trunk may be arranged in the following groups, viz. cervical 8, thoracic 12, lumbar 5, sacral 5, and coccygeal from 5 to 8. Those of the occipital region of the head are usually described as being 4 in number. In mammals, primitive segments of the head can be recognized only in the occipital region, but a study of the lower vertebrates leads to the belief that they are present also in the anterior part of the head, and that altogether 9 segments are represented in the cephalic region. Part 9. Separation of the embryo. The embryo increases rapidly in size, but the circumference of the embryonic disc, or line of meeting of the embryonic and amniotic parts of the ectoderm, is of relatively slow growth, and gradually comes to form a constriction between the embryo and the greater part of the yolk sac. By means of this constriction, which corresponds to the future umbilicus, a small part of the yolk sac is enclosed within the embryo and constitutes the primitive digestive tube. The embryo increases more rapidly in length than in width, and its cephalic and caudal ends soon extend beyond the corresponding parts of the circumference of the embryonic disc, and are bent in a ventral direction to form the cephalic and caudal folds respectively. The cephalic fold is first formed, and as the proamniotic area, lying immediately in front of the pericardial area, forms the anterior limit of the circumference of the embryonic disc, the forward growth of the head necessarily carries with it the posterior end of the pericardial area, so that this area and the buccopharyngeal membrane are folded back under the head of the embryo, which now encloses a diverticulum of the yolk sac named the foregut. The caudal end of the embryo is at first connected to the chorion by a band of mesoderm called the body stalk, but with the formation of the caudal fold, the body stalk assumes a ventral position. A diverticulum of the yolk sac extends into the tail fold and is termed the hindgut. Between the foregut and the hindgut there exists for a time a wide opening into the yolk sac, but the latter is gradually reduced to a small pear-shaped sac, sometimes termed the umbilical vesicle, and the channel of communication is at the same time narrowed and elongated to form a tube called the vitelline duct. Part 10. The yolk sac. The yolk sac is situated on the ventral aspect of the embryo. It is lined by entoderm, outside of which is a layer of mesoderm. It is filled with fluid, the vitelline fluid, which possibly may be utilized for the nourishment of the embryo during the earlier stages of its existence. Blood is conveyed to the wall of the sac by the primitive aortae, and after circulating through a wide-meshed capillary plexus, is returned by the vitelline veins to the tubular heart of the embryo. This constitutes the vitelline circulation, and by means of it nutritive material is absorbed from the yolk sac and conveyed to the embryo. At the end of the 4th week, the yolk sac presents the appearance of a small pear-shaped vesicle, umbilical vesicle, opening into the digestive tube by a long narrow tube, the vitelline duct. The vesicle can be seen in the afterbirth as a small, somewhat oval-shaped body whose diameter varies from 1 mm to 5 mm. It is situated between the amnion and the chorion, and may lie on or at a varying distance from the placenta. As a rule, the duct undergoes complete obliteration during the 7th week, but in about 3% of cases its proximal part persists as a diverticulum from the small intestine. Meckel's diverticulum, which is situated about 3 or 4 feet above the ileocolic junction, and may be attached by a fibrous cord to the abdominal wall at the umbilicus. Sometimes a narrowing of the lumen of the ileum is seen opposite the site of the attachment of the duct. End of Section 5. Recording by Leanne Howlett. Section 6 of Gray's Anatomy, Part 1. Anatomy of the Human Body, Part 1, by Henry Gray. Part 11. Development of the fetal membranes and placenta. The allantois arises as a tubular diverticulum of the posterior part of the yolk sac. When the hindgut is developed, the allantois is carried backward with it and then opens into the cloaca, or terminal part of the hindgut. It grows out into the body stalk, a mass of mesoderm which lies below and around the tail end of the embryo. The diverticulum is lined by entoderm and covered by mesoderm, and in the latter are carried the allantoic or umbilical vessels. In reptiles, birds, and many mammals, the allantois becomes expanded into a vesicle which projects into the extraembryonic coelom. If its further development be traced in the bird, it is seen to project to the right side of the embryo, and gradually expanding, it spreads over its dorsal surface as a flattened sac between the amnion and the serosa, and extending in all directions, ultimately surrounds the yolk. Its outer wall becomes applied to and fuses with the serosa, which lies immediately inside the shell membrane. Blood is carried to the allantoic sac by the two allantoic or umbilical arteries, which are continuous with the primitive aortae, and after circulating through the allantoic capillaries is returned to the primitive heart by the two umbilical veins. In this way the allantoic circulation, which is of the utmost importance in connection with the respiration and nutrition of the chick, is established. Oxygen is taken from and carbonic acid is given up to the atmosphere through the eggshell, while nutritive materials are at the same time absorbed by the blood from the yolk. In man and other primates, the nature of the allantois is entirely different from that just described. Here it exists merely as a narrow, tubular diverticulum of the hindgut, and never assumes the form of a vesicle outside the embryo. With the formation of the amnion, the embryo is, in most animals, entirely separated from the chorion, and is only again united to it when the allantoic mesoderm spreads over and becomes applied to its inner surface. The human embryo, on the other hand, as was pointed out by His, is never wholly separated from the chorion, its tail end being from the first connected with the chorion by means of a thick band of mesoderm named the body stalk. Into this stalk the tube of the allantois extends. The amnion. The amnion is a membranous sac which surrounds and protects the embryo. It is developed in reptiles, birds, and mammals, which are hence called amniota, but not in amphibia and fishes, which are consequently termed anamnia. In the human embryo the earliest stages of the formation of the amnion have not been observed. In the youngest embryo which has been studied, the amnion was already present as a closed sac, and is indicated on page 46, appears in the inner cell mass as a cavity. This cavity is roofed in by a single stratum of flattened ectodermal cells, the amniotic ectoderm, and its floor consists of the prismatic ectoderm of the embryonic disc, the continuity between the roof and floor being established at the margin of the embryonic disc. Outside the amniotic ectoderm is a thin layer of mesoderm, which is continuous with that of the soma to pleur and is connected by the body stalk with the mesodermal lining of the chorion. When first formed, the amnion is in contact with the body of the embryo, but about the 4th or 5th week, fluid, liquor amnii, begins to accumulate within it. This fluid increases in quantity and causes the amnion to expand and ultimately to adhere to the inner surface of the chorion, so that the extra-embryonic part of the coelom is obliterated. The liquor amnii increases in quantity up to the 6th or 7th month of pregnancy, after which it diminishes somewhat. At the end of pregnancy it amounts to about 1 liter. It allows of the free movements of the fetus during the later stages of pregnancy, and also protects it by diminishing the risk of injury from without. It contains less than 2% of solids, consisting of urea and other extractives, inorganic salts, a small amount of protein, and frequently a trace of sugar. That some of the liquor amnii is swallowed by the fetus is proved by the fact that epidermal debris and hairs have been found among the contents of the fetal alimentary canal. In reptiles, birds, and many mammals the amnion is developed in the following manner. At the point of constriction where the primitive digestive tube of the embryo joins the yolk sac, a reflection or folding upward of the soma to pleur takes place. This, the amniotic fold, first makes its appearance at the cephalic extremity, and subsequently at the caudal end and sides of the embryo, and gradually rising more and more, its different parts meet and fuse over the dorsal aspect of the embryo, and enclose a cavity, the amniotic cavity. After the fusion of the edges of the amniotic fold, the two layers of the fold become completely separated, the inner forming the amnion, the outer the false amnion, or serosa. The space between the amnion and the serosa constitutes the extra-embryonic coelom, and for a time communicates with the embryonic coelom. The umbilical cord and body stalk. The umbilical cord attaches the fetus to the placenta. Its length at full time, as a rule, is about equal to the length of the fetus, that is, about 50 centimeters, but it may be greatly diminished or increased. The rudiment of the umbilical cord is represented by the tissue which connects the rapidly growing embryo with the extra-embryonic area of the ovum. Included in this tissue are the body stalk and the vitelline duct, the former containing the allantoic diverticulum and the umbilical vessels, the latter forming the communication between the digestive tube and the yolk sac. The body stalk is the posterior segment of the embryonic area, and is attached to the chorion. It consists of a plate of mesoderm covered by thickened ectoderm, on which a trace of the neural groove can be seen, indicating its continuity with the embryo. Running through its mesoderm are the 2 umbilical arteries and the 2 umbilical veins, together with the canal of the allantois, the last being lined by entoderm. Its dorsal surface is covered by the amnion, while its ventral surface is bounded by the extra-embryonic coelom and is in contact with the vitelline duct and yolk sac. With the rapid elongation of the embryo and the formation of the tail fold, the body stalk comes to lie on the ventral surface of the embryo, where its mesoderm blends with that of the yolk sac and the vitelline duct. The lateral leaves of somatopleur then grow round on each side, and, meeting on the ventral aspect of the allantois, enclose the vitelline duct and vessels, together with a part of the extra-embryonic coelom. The latter is ultimately obliterated. The cord is covered by a layer of ectoderm which is continuous with that of the amnion, and its various constituents are enveloped by embryonic gelatinous tissue, jelly of Wharton. The vitelline vessels and duct, together with the right umbilical vein, undergo atrophy and disappear, and thus the cord, at birth, contains a pair of umbilical arteries and one, the left, umbilical vein. Implantation or embedding of the ovum. As described, fertilization of the ovum occurs in the lateral or ampullary end of the uterine tube and is immediately followed by segmentation. On reaching the cavity of the uterus, the segmented ovum adheres like a parasite to the uterine mucous membrane, destroys the epithelium over the area of contact, and excavates for itself a cavity in the mucous membrane in which it becomes embedded. In the ovum described by Bryce and Teacher, the point of entrance was visible as a small gap closed by a mass of fibrin and leukocytes. In the ovum described by Peters, the opening was covered by a mushroom-shaped mass of fibrin and blood clot, the narrow stalk of which plugged the aperture in the mucous membrane. Soon, however, all trace of the opening is lost, and the ovum is then completely surrounded by the uterine mucous membrane. The structure actively concerned in the process of excavation is the trophoblast of the ovum, which possesses the power of dissolving and absorbing the uterine tissues. The trophoblast proliferates rapidly and forms a network of branching processes which cover the entire ovum and invade and destroy the maternal tissues and open into the maternal blood vessels, with the result that the spaces in the trophoblastic network are filled with maternal blood. These spaces communicate freely with one another and become greatly distended and form the intervillous space. The decidua. Before the fertilized ovum reaches the uterus, the mucous membrane of the body of the uterus undergoes important changes, and is then known as the decidua. The thickness and vascularity of the mucous membrane are greatly increased. Its glands are elongated and open on its free surface by funnel-shaped orifices, while their deeper portions are tortuous and dilated into irregular spaces. The interglandular tissue is also increased in quantity, and is crowded with large, round, oval, or polygonal cells termed decidual cells. These changes are well advanced by the second month of pregnancy, when the mucous membrane consists of the following strata. 1. Stratum compactum. Next, the free surface. In this the uterine glands are only slightly expanded and are lined by columnar cells. 2. Stratum spongiosum, in which the gland tubes are greatly dilated and very tortuous, and are ultimately separated from one another by only a small amount of interglandular tissue, while their lining cells are flattened or cubical. 3. A thin, unaltered or boundary layer, next the uterine muscular fibers, containing the deepest parts of the uterine glands, which are not dilated, and are lined with columnar epithelium. It is from this epithelium that the epithelial lining of the uterus is regenerated after pregnancy. Distinctive names are applied to different portions of the decidua. The part which covers in the ovum is named the decidua capsularis. The portion which intervenes between the ovum and the uterine wall is named the decidua basalis, or decidua placentalis. It is here that the placenta is subsequently developed. The part of the decidua which lines the remainder of the body of the uterus is known as the decidua vira, or decidua parietalis. Coincidentally with the growth of the embryo, the decidua capsularis is thinned and extended, and the space between it and the decidua vira is gradually obliterated, so that by the 3rd month of pregnancy the two are in contact. By the 5th month of pregnancy, the decidua capsularis has practically disappeared, while during the succeeding months the decidua vira also undergoes atrophy, owing to the increased pressure. The glands of the stratum compactum are obliterated, and their epithelium is lost. In the stratum spongiosum, the glands are compressed and appear as slit-like fissures, while their epithelium undergoes degeneration. In the unaltered or boundary layer, however, the glandular epithelium retains a columnar or cubical form. The chorion. The chorion consists of 2 layers, an outer formed by the primitive ectoderm or trophoblast, and an inner by the somatic mesoderm. With this latter the amnion is in contact. The trophoblast is made up of an internal layer of cubical or prismatic cells, the cytotrophoblast, or layer of Langhans, and an external layer of richly nucleated protoplasm devoid of cell boundaries, the syncytiotrophoblast. It undergoes rapid proliferation and forms numerous processes: the chorionic villi, which invade and destroy the uterine decidua and at the same time absorb from it nutritive materials for the growth of the embryo. The chorionic villi are at first small and non-vascular, and consist of trophoblast only, but they increase in size and ramify, while the mesoderm, carrying branches of the umbilical vessels, grows into them, and in this way they are vascularized. Blood is carried to the villi by the branches of the umbilical arteries, and after circulating through the capillaries of the villi, is returned to the embryo by the umbilical veins. Until about the end of the 2nd month of pregnancy, the villi cover the entire chorion and are almost uniform in size, but after this they develop unequally. The greater part of the chorion is in contact with the decidua capsularis, and over this portion of the villi, with their contained vessels, undergo atrophy, so that by the 4th month scarcely a trace of them is left, and hence this part of the chorion becomes smooth, and is named the chorion live. As it takes no share in the formation of the placenta, it is also named the non-placental part of the chorion. On the other hand, the villi on that part of the chorion which is in contact with the decidua placentalis increase greatly in size and complexity, and hence this part is named the chorion frondosum. The placenta. The placenta connects the fetus to the uterine wall, and is the organ by means of which the nutritive, respiratory, and excretory functions of the fetus are carried out. It is composed of fetal and maternal portions. Fetal portion. The fetal portion of the placenta consists of the villi of the chorion frondosum. These branch repeatedly and increase enormously in size. These greatly ramified villi are suspended in the intervillous space, and are bathed in maternal blood, which is conveyed to the space by the uterine arteries and carried away by the uterine veins. A branch of an umbilical artery enters each villus and ends in a capillary plexus, from which the blood is drained by a tributary of the umbilical vein. The vessels of the villus are surrounded by a thin layer of mesoderm, consisting of gelatinous connective tissue, which is covered by 2 strata of ectodermal cells derived from the trophoblast. The deeper stratum, next the mesodermic tissue, represents the cytotrophoblast or layer of Langhans. The superficial, in contact with the maternal blood, the syncytiotrophoblast. After the 5th month the two strata of cells are replaced by a single layer of somewhat flattened cells. Maternal portion. The maternal portion of the placenta is formed by the decidua placentalis containing the intervillous space. As already explained, this space is produced by the enlargement and intercommunication of the spaces in the trophoblastic network. The changes involve the disappearance of the greater portion of the stratum compactum, but the deeper part of this layer persists and is condensed to form what is known as the basal plate. Between this plate and the uterine muscular fibers are the stratum spongiosum and the boundary layer. Through these and the basal plate, the uterine arteries and veins pass to and from the intervillous space. The endothelial lining of the uterine vessels ceases at the point where they terminate in the intervillous space, which is lined by the syncytio trophoblast. Portions of the stratum compactum persist and are condensed to form a series of septa, which extend from the basal plate through the thickness of the placenta and subdivide it into the lobules or cotyledons seen on the uterine surface of the detached placenta. The fetal and maternal blood currents traverse the placenta, the former passing through the blood vessels of the placental villi, and the latter through the intervillous space. The two currents do not intermingle, being separated from each other by the delicate walls of the villi. Nevertheless, the fetal blood is able to absorb, through the walls of the villi, oxygen and nutritive materials from the maternal blood, and give up to the latter its waste products. The blood, so purified, is carried back to the fetus by the umbilical vein. It will thus be seen that the placenta not only establishes a mechanical connection between the mother and the fetus, but subserves for the latter the purposes of nutrition, respiration, and excretion. In favor of the view that the placenta possesses certain selective powers may be mentioned the fact that glucose is more plentiful in the maternal than in the fetal blood. It is interesting to note also that the proportion of iron and of lime and potash in the fetus is increased during the last months of pregnancy. Further, there is evidence that the maternal leukocytes may migrate into the fetal blood, since leukocytes are much more numerous in the blood of the umbilical vein than in that of the umbilical arteries. The placenta is usually attached near the fundus uteri, and more frequently on the posterior than on the anterior wall of the uterus. It may, however, occupy a lower position, and in rare cases its site is close to the orificium internum uteri, which it may occlude, thus giving rise to the condition known as placenta previa. Separation of the placenta. After the child is born, the placenta and membranes are expelled from the uterus as the afterbirth. The separation of the placenta from the uterine wall takes place through the stratum spongiosum and necessarily causes rupture of the uterine vessels. The orifices of the torn vessels are, however, closed by the firm contraction of the uterine muscular fibers, and thus postpartum hemorrhage is controlled. The epithelial lining of the uterus is regenerated by the proliferation and extension of the epithelium, which lines the persistent portions of the uterine glands in the unaltered layer of the decidua. The expelled placenta appears as a discoid mass which weighs about 450 grams and has a diameter of from 15 to 20 centimeters. Its average thickness is about 3 centimeters, but this diminishes rapidly toward the circumference of the disc, which is continuous with the membranes. Its uterine surface is divided by a series of fissures into lobules or cotyledons, the fissures containing the remains of the septa, which extended between the maternal and fetal portions. Most of these septa end in irregular or pointed processes. Others, especially those near the edge of the placenta, pass through its thickness and are attached to the chorion. In the early months these septa convey branches of the uterine arteries which open into the intervillous space on the surfaces of the septa. The fetal surface of the placenta is smooth, being closely invested by the amnion. Seen through the latter, the chorion presents a mottled appearance, consisting of gray, purple, or yellowish areas. The umbilical cord is usually attached near the center of the placenta, but may be inserted anywhere between the center and the margin. In some cases it is inserted into the membranes, that is, the filamentous insertion. From the attachment of the cord, the longer branches of the umbilical vessels radiate under the amnion, the veins being deeper and larger than the arteries. The remains of the vitelline duct and yolk sac may be sometimes observed beneath the amnion, close to the cord, the former as an attenuated thread, the latter as a minute sac. On section, the placenta presents a soft, spongy appearance, caused by the greatly branched villi. Surrounding them is a varying amount of maternal blood, giving the dark red color to the placenta. Many of the larger villi extend from the chorionic to the decidual surface, while others are attached to the septa, which separate the cotyledons, but the great majority of the villi hang free in the intervillous space. End of Section 6. Recording by Leanne Howlett. Section 7 of Gray's Anatomy, Part 1. Anatomy of the Human Body, Part 1, by Henry Gray. Part 12. The Branchial Region. The branchial or visceral arches and pharyngeal pouches. In the lateral walls of the anterior part of the foregut, 5 pharyngeal pouches appear. Each of the upper 4 pouches is prolonged into a dorsal and a ventral diverticulum. Over these pouches corresponding indentations of the ectoderm occur, forming what are known as the branchial or outer pharyngeal grooves. The intervening mesoderm is pressed aside and the ectoderm comes for a time into contact with the entodermal lining of the foregut, and the 2 layers unite along the floors of the grooves to form thin closing membranes between the foregut and the exterior. Later, the mesoderm again penetrates between the entoderm and the ectoderm. In gill-bearing animals, the closing membranes disappear and the grooves become complete clefts, the gill clefts, opening from the pharynx onto the exterior. Perforation, however, does not occur in birds or mammals. The grooves separate a series of rounded bars or arches, the branchial or visceral arches, in which thickening of the mesoderm takes place. The dorsal ends of these arches are attached to the sides of the head, while the ventral extremities ultimately meet in the middle line of the neck. In all, 6 arches make their appearance, but of these only the first 4 are visible externally. The first arch is named the mandibular, and the second the hyoid. The others have no distinctive names. In each arch a cartilaginous bar consisting of right and left halves is developed, and with each of these there is one of the primitive aortic arches. The mandibular arch lies between the first branchial groove and the stomotium. From it are developed the lower lip, the mandible, the muscles of mastication, and the anterior part of the tongue. Its cartilaginous bar is formed by what are known as Meckel's cartilages. Above this the incus is developed. The dorsal end of each cartilage is connected with the ear capsule and is ossified to form the malleus. The ventral ends meet each other in the region of the symphysis mentae, and are usually regarded as undergoing ossification to form that portion of the mandible which contains the incisor teeth. The intervening part of the cartilage disappears. The portion immediately adjacent to the malleus is replaced by fibrous membrane, which constitutes the sphenomandibular ligament, while from the connective tissue covering the remainder of the cartilage, the greater part of the mandible is ossified. From the dorsal ends of the mandibular arch a triangular process, the maxillary process, grows forward on either side and forms the cheek and lateral part of the upper lip. The second or hyoid arch assists in forming the side and front of the neck. From its cartilage are developed the styloid process, stylohyoid ligament, and lesser cornu of the hyoid bone. The stages probably arises in the upper part of this arch. The cartilage of the third arch gives origin to the greater cornu of the hyoid bone. The ventral ends of the second and third arches unite with those of the opposite side, and form a transverse band, from which the body of the hyoid bone and the posterior part of the tongue are developed. The ventral portions of the cartilages of the fourth and fifth arches unite to form the thyroid cartilage. From the cartilages of the sixth arch, the cricoid and arytenoid cartilages and the cartilages of the trachea are developed. The mandibular and hyoid arches grow more rapidly than those behind them, with the result that the latter become, to a certain extent, telescoped within the former, and a deep depression, the sinus cervicalis, is formed on either side of the neck. This sinus is bounded in front by the hyoid arch, and behind by the thoracic wall. It is ultimately obliterated by the fusion of its walls. From the first branchial groove, the concha auriculae and external acoustic meatus are developed, while around the groove there appear on the mandibular and hyoid arches a number of swellings from which the auricula or pinna is formed. The first pharyngeal pouch is prolonged dorsally to form the auditory tube and the tympanic cavity. The closing membrane between the mandibular and hyoid arches is invaded by mesoderm and forms the tympanic membrane. No traces of the second, third, and fourth branchial grooves persist. The inner part of the second pharyngeal pouch is named the sinus tonsillaris. In it the tonsil is developed, above which a trace of the sinus persists as the supratonsillar fossa. The fossa of Rosenmuller, or lateral recess of the pharynx, is by some regarded as a persistent part of the second pharyngeal pouch, but it is probably developed as a secondary formation. From the third pharyngeal pouch the thymus arises as an entodermal diverticulum on either side, and from the fourth pouches small diverticula project and become incorporated with the thymus. But in man these diverticula probably never form true thymus tissue. The parathyroids also arise as diverticula from the third and fourth pouches. From the fifth pouches the ultimobranchial bodies originate and are enveloped by the lateral prolongations of the median thyroid rudiment. They do not, however, form true thyroid tissue, nor are any traces of them found in the human adult. The nose and face. During the third week, two areas of thickened ectoderm, the olfactory areas, appear immediately under the forebrain in the anterior wall of the stomodium, one on either side of a region termed the frontonasal process. By the upgrowth of the surrounding parts, these areas are converted into pits, the olfactory pits, which indent the frontonasal process and divide it into a medial and two lateral nasal processes. The rounded lateral angles of the medial process constitute the globular processes of His. The olfactory pits form the rudiments of the nasal cavities, and from their ectodermal lining the epithelium of the nasal cavities, with the exception of that of the inferior metices, is derived. The globular processes are prolonged backward as plates, termed the nasal laminae. These laminae are at first some distance apart, but gradually approaching, they ultimately fuse and form the nasal septum. The processes themselves meet in the middle line and form the premaxillae and the philtrum or central part of the upper lip. The depressed part of the medial nasal process between the globular processes forms the lower part of the nasal septum or columella, while above this is seen a prominent angle, which becomes the future apex, and still higher a flat area, the future bridge, of the nose. The lateral nasal processes form the alee of the nose. Continuous with the dorsal end of the mandibular arch, and growing forward from its cephalic border, is a triangular process, the maxillary process, the ventral extremity of which is separated from the mandibular arch by a shaped notch. The maxillary process forms the lateral wall and floor of the orbit, and in it are ossified the zygomatic bone and the greater part of the maxilla. It meets with the lateral nasal process, from which, however, it is separated for a time by a groove, the naso-optic furrow, that extends from the furrow encircling the eyeball to the olfactory pit. The maxillary processes ultimately fuse with the lateral nasal and globular processes, and form the lateral parts of the upper lip and the posterior boundaries of the nares. From the third to the fifth month the nares are filled by masses of epithelium, on the breaking down and disappearance of which the permanent openings are produced. The maxillary process also gives rise to the lower portion of the lateral wall of the nasal cavity. The roof of the nose and the remaining parts of the lateral wall, viz., the ethmoidal labyrinth, the inferior nasal concha, the lateral cartilage, and the lateral crus of the alar cartilage, are developed in the lateral nasal process. By the fusion of the maxillary and nasal processes in the roof of the stomodium, the primitive palate is formed, and the olfactory pits extend backward above it. The posterior end of each pit is closed by an epithelial membrane, the bucco-nasal membrane, formed by the apposition of the nasal and stomodial epithelium. By the rupture of these membranes, the primitive choanae, or openings between the olfactory pits and the stomodium, are established. The floor of the nasal cavity is completed by the development of a pair of shelf-like palatine processes, which extend medialward from the maxillary processes. These coalesce with each other in the middle line, and constitute the entire palate, except a small part in front which is formed by the premaxillary bones. Two apertures persist for a time between the palatine processes and the premaxillae, and represent the permanent channels which in the lower animals connect the nose and mouth. The union of the parts which form the palate commences in front, the premaxillary and palatine processes joining in the 8th week, while the region of the future hard palate is completed by the 9th, and that of the soft palate by the 11th week. By the completion of the palate, the permanent choanae are formed, and are situated a considerable distance behind the primitive choanae. The deformity known as cleft palate results from a non-union of the palatine processes, and that of hare lip through a non-union of the maxillary and globular processes. See page 199. The nasal cavity becomes divided by a vertical septum, which extends downward and backward from the medial nasal process and nasal laminae, and unites below with the palatine processes. Into this septum a plate of cartilage extends from the under aspect of the ethmoid plate of the chondrocranium. The anterior part of this cartilaginous plate persists as the septal cartilage of the nose and the medial crus of the alar cartilage, but the posterior and upper parts are replaced by the vomer and perpendicular plate of the ethmoid. On either side of the nasal septum, at its lower and anterior part, the ectoderm is invaginated to form a blind pouch or diverticulum, which extends backward and upward into the nasal septum, and is supported by a curved plate of cartilage. These pouches form the rudiments of the vomeronasal organs of Jacobson, which open below, close to the junction of the premaxillary and maxillary bones. The limbs. The limbs begin to make their appearance in the 3rd week as small elevations or buds at the side of the trunk. Prolongations from the muscle and cutis plates of several primitive segments extend into each bud, and carry with them the anterior divisions of the corresponding spinal nerves. The nerves supplying the limbs indicate the number of primitive segments which contribute to their formation, the upper limb being derived from 7, viz., 4th cervical to 2nd thoracic inclusive, and the lower limb from 10, viz., 12th thoracic to 4th sacral inclusive. The axial part of the mesoderm of the limb bud becomes condensed and converted into its cartilaginous skeleton, and by the ossification of this, the bones of the limbs are formed. By the 6th week the 3 chief divisions of the limbs are marked off by furrows, the upper into arm, forearm, and hand, the lower into thigh, leg, and foot. The limbs are at first directed backward nearly parallel to the long axis of the trunk, and each presents 2 surfaces and 2 borders. Of the surfaces, one, the future flexor surface of the limb, is directed ventrally; the other, the extensor surface, dorsally. One border, the preaxial, looks forward toward the cephalic end of the embryo, and the other, the postaxial, backward toward the caudal end. The lateral epicondyle of the humerus, the radius, and the thumb lie along the preaxial border of the upper limb, and the medial epicondyle of the femur, the tibia, and the great toe along the corresponding border of the lower limb. The preaxial part is derived from the anterior segments, the postaxial from the posterior segments of the limb bud, and this explains to a large extent the innervation of the adult limb, the nerves of the more anterior segments being distributed along the preaxial, radial or tibial, and those of the more posterior along the postaxial, ulnar or fibular border of the limb. The limbs next undergo a rotation or torsion through an angle of 90 degrees around their long axes, the rotation being effected almost entirely at the limb girdles. In the upper limb the rotation is outward and forward; in the lower limb, inward and backward. As a consequence of this rotation, the preaxial, radial border of the forelimb is directed lateralward, and the preaxial, tibial border of the hind limb is directed medialward. Thus, the flexor surface of the forelimb is turned forward, and that of the hind limb backward. End of Section 7. Recording by Leanne Howlett.
Section 8 of Gray's Anatomy, Part 1. Anatomy of the Human Body, Part 1, by Henry Gray. Development of the body cavities. In the human embryo described by Peters, the mesoderm outside the embryonic disc is split into two layers, enclosing an extra-embryonic column. There is no trace of an intra-embryonic column. At a later stage, 4 cavities are formed within the embryo. These, one on either side, within the mesoderm of the pericardial area, and one in either lateral mass of the general mesoderm. All these are at first independent of each other and of the extra-embryonic coelom, but later they become continuous. The 2 cavities in the general mesoderm unite on the ventral aspect of the gut and form the pleuroperitoneal cavity, which becomes continuous with the remains of the extra-embryonic coelom around the umbilicus. The 2 cavities in the pericardial area rapidly join to form a single pericardial cavity, and this from 2 lateral diverticula extend calderward to open into the pleuroperitoneal cavity. Between the 2 latter diverticula is a mass of mesoderm containing the ducts of Cuvier, and this is continuous ventrally with the mesoderm, in which the umbilical veins are passing to the sinus venosus. A septum of mesoderm thus extends across the body of the embryo. It is attached in front to the body wall, between the pericardium and umbilicus, behind the body wall at the level of the second cervical segment. Laterally it is deficient where the pericardial pleuroperitoneal cavities communicate, while it is perforated in the middle line by the foregut. This partition is termed septum transversum, and is at first a bulky plate of tissue. As development proceeds, the dorsal end of the septum is carried gradually calderward, and when it reaches the 5th cervical segment, muscular tissue with the phrenic nerve grows into it. It continues to recede, however, until it reaches the position of the adult diaphragm on the bodies of the upper lumbar vertebrae. The liver buds grow into the septum transversum and undergo development there. The lung buds, meantime, have grown out from the foregut and project laterally into the forepart of the pleuroperitoneal cavity. The developing stomach and liver are embedded in the septum transversum. Caudal to this, the intestines project into the back part of the pleuroperitoneal cavity. Owing to the descent of the dorsal end of the septum transversum, the lung buds come to lie above the septum, and thus pleural and peritoneal portions of the pleuroperitoneal cavity, still, however, in free communication with one another, may be recognized. The pericardial cavity opens into the pleural part. The ultimate separation of the permanent cavities from one another is effected by the growth of a ridge of tissue on either side of the mesoderm surrounding the duct of Cuvier. The front part of this ridge grows across and obliterates the pleuropericardial opening. The hinder part grows across the pleuroperitoneal opening. With the continued growth of the lungs, the pleural cavities are pushed forward in the body wall towards the ventral median line, thus separating the pericardium from the lateral thoracic walls. The further development of the peritoneal cavity has been described with the development of the digestive tube. The form of the embryo at different stages of its growth. First week. During this period the ovum is in the uterine tube. Having been fertilized in the upper part of the tube, it slowly passes down, undergoing segmentation, and reaches the uterus. Peters describes a specimen, the age of which he reckoned as from 3 to 4 days. Footnote. Bryce and Teacher, Early Development and Embedding of the Human Ovum, 1908, have described an ovum which they regard as 13 to 14 days old. In it, the two vesicles, the amnion and yolk sac, were present, but there was no trace of a layer of embryonic ectoderm. They are of opinion that the age of Peter's ovum has been understated, and estimate it as between 13 1/2 and 14 1/2 days. End footnote. It was embedded in the decidua on the posterior wall of the uterus and enveloped by a decidua capsularis, the central part of which, however, consisted merely of a layer of fibrin. The ovum was in the form of a sac, the outer wall of which consisted of a layer of trophoblast. Inside this was a thin layer of mesoderm composed of round, oval and spindle-shaped cells. Numerous villous processes, some consisting of trophoblast only, others possessing a core of mesoderm, projected from the surface of the ovum into the surrounding decidua. Inside this sac the rudiment of the embryo was found in the form of a patch of ectoderm, covered by a small but completely closed amnion. It possessed a minute yolk sac and was surrounded by mesoderm, which was connected by a band to that lining the trophoblast. Second week. By the end of this week the ovum has increased considerably in size, and the majority of its villi are vascularized. The embryo has assumed a definite form, and its cephalic and caudal extremities are easily distinguished. The neural folds are partly united. The embryo is more completely separated from the yolk sac, and the paraxial mesoderm is being divided into the primitive segments. Third week. By the end of the third week the embryo is strongly curved, and the primitive segments number about 30. The primary divisions of the brain are visible, and the optic and auditory vesicles are formed. 4 branchial grooves are present. The stomadeum is well marked, and the bucopharyngeal membrane has disappeared. The rudiments of the limbs are seen as short buds, and the Wolffian bodies are visible. Fourth week. The embryo is markedly curved on itself, and when viewed in profile is almost circular in outline. The cerebral hemispheres appear as hollow buds, and the elevations which form the rudiments of the auricula are visible. The limbs now appear as oval, flattened projections. Fifth week. The embryo is less curved, and the head is relatively of large size. Differentiation of the limbs into their segments occurs. The nose forms a short, flattened projection. The colloquial tubercle is evident. Sixth week. The curvature of the embryo is further diminished. The branchial grooves, except the first, have disappeared, and the rudiments of the fingers and toes can be recognized. Seventh and eighth weeks. The flexure of the head is gradually reduced, and the neck is somewhat lengthened. The upper lip is completed, and the nose is more prominent. The nostrils are directed forward, and the palate is not completely developed. The eyelids are present in the shape of folds above and below the eye, and the different parts of the auricula are distinguishable. By the end of the second month the fetus measures from 28 to 30 millimeters in length. Third month. The head is extended and the neck is lengthened. The eyelids meet and fuse, remaining closed until the end of the 6th month. The limbs are well developed and nails appear on the digits. The external generative organs are so far differentiated that it is possible to distinguish the sex. By the end of this month the length of the fetus is about 7 centimeters, but if the legs be included it is from 9 to 10 centimeters. Fourth month. The loop of gut which projected into the umbilical cord is withdrawn within the fetus. The hairs begin to make their appearance. There is a general increase in size so that by the end of the 4th month the fetus is from 12 to 13 centimeters in length, but if the legs be included it is from 16 to 20 centimeters. 5th month. It is during this month that the first movements of the fetus are usually observed. The eruption of hair on the head commences, and the vernix caseosa begins to be deposited. By the end of this month the total length of the fetus, including the legs, is from 25 to 27 centimeters. 6th month. The body is covered by fine hairs, lanugo, and the deposit of vernix caseosa is considerable. The papillae of the skin are developed, and the free border of the nail projects from the corium of the dermis. Measured from vertex to heels, the total length of the fetus at the end of this month is from 30 to 32 centimeters. 7th month. The pupillary membrane atrophies and the eyelids are open. The testes descends with the vaginal sac of the peritoneum. From vertex to heels, the total length at the end of the 7th month is from 35 to 36 centimeters. The weight is a little over 3 pounds. 8th month. The skin assumes a pink color and is now entirely coated with vernix caseosa, and the langua begins to disappear. Subcutaneous fat has been developed to a considerable extent, and the fetus presents a plump appearance. The total length, that is, from head to heels, at the end of the 8th month is about 40 centimeters, and the weight varies between 4 1/2 and 5 1/2 pounds. 9th month. The langua has largely disappeared from the trunk. The umbilicus is almost in the middle of the body, and the testes are in the scrotum. At full time the fetus weighs from 6 1/2 to 8 pounds, and measures from head to heels about 50 centimeters. End of section 8.
Section 9 of Gray's Anatomy, Part 1. Anatomy of the Human Body, Part 1, by Henry Gray. Osteology. Introduction. Development of the skeleton. The general framework of the body is built up mainly of a series of bones, supplemented, however, in certain regions by pieces of cartilage. The bony part of the framework constitutes the skeleton. In the skeleton of the adult there are 206 distinct bones, as follows. The axial skeleton, divided into the vertebral column with 26 bones, the skull with 22 bones, the hyoid bone 1, the ribs and sternum 25, 74 bones in total. The appendicular skeleton, upper extremities with 64 bones, lower extremities with 62 bones, 126 altogether, and the auditory ossicles 6, total of 206. The patellae are included in this enumeration, but the smaller sesamoid bones are not reckoned. Bones are divisible into 4 classes: long, short, flat, and irregular. Long bones. The long bones are found in the limbs, and each consists of a body or shaft and 2 extremities. The body, or diaphysis, is cylindrical, with a central cavity termed the medullary canal. The wall consists of dense, compact tissue of considerable thickness in the middle part of the body, but becoming thinner toward the extremities. Within the medullary canal is some cancellous tissue, scanty in the middle of the body, but greater in amount towards the ends. The extremities are generally expanded for the purpose of articulation and to afford broad surfaces for muscular attachment. They are usually developed from separate centres of ossification termed epiphyses, and consist of cancellous tissue surrounded by thin compact bone. The medullary canal and the spaces in the cancellous tissue are filled with marrow. The long bones are not straight, but curved, the curve generally taking place in 2 planes, thus affording greater strength to the bone. The bones belonging to this class are the clavicle, humerus, radius, ulna, femur, tibia, fibula, metacarpals, metatarsals, and phalanges. Short bones. Where a part of the skeleton is intended for strength and compactness combined with limited movement, it is constructed of a number of short bones, as in the carpus and tarsus. These consist of cancellous tissue covered by a thin crust of compact substance. The patellae, together with the other sesamoid bones, are by some regarded as short bones. Flat bones. Where the principal requirement is either extensive protection or the provision of broad surfaces for muscular attachment, the bones are expanded into broad, flat plates, as in the skull and the scapula. These bones are composed of two thin layers of compact tissue enclosing between them a variable quantity of cancellous tissue. In the cranial bones, the layers of compact tissue are familiarly known as the tables of the skull. The outer one is thick and tough, the inner is thin, dense, and brittle, and hence is termed the vitreous table. The intervening cancellous tissue is called the diploe, and this, in certain regions of the skull, becomes absorbed so as to leave spaces filled with air sinuses, between the two tables. The flat bones are the occipital, parietal, frontal, nasal, lacrimal, vomer, scapular, os cocci, hip bone, sternum, ribs, and, according to some, the patella. Irregular bones. The irregular bones are such as, from their peculiar form, cannot be grouped under the preceding heads. They consist of cancellous tissue enclosed within a thin layer of compact bone. The irregular bones are the vertebrae, sacrum, coccyx, temporal, sphenoid, ethmoid, zygomatic, maxilla, mandible, palatine, inferior nasal concha, and hyoid. Surfaces of bones. If the surface of a bone be examined, certain eminences and depressions are seen. These eminences and depressions are of two kinds, articular and non-articular. Well-marked examples of articular eminences are found in the heads of the humerus and femur, and of articular depressions in the glenoid cavity of the scapula and the acetabulum of the hip bone. Non-articular eminences are designated according to their form. Thus a broad, rough, uneven elevation is called a tuberosity, protuberance, or process; a small, rough prominence, a tubercle; a sharp, slender, pointed eminence, a spine; a narrow, rough elevation, running some way along the surface, a ridge, crest, or line. Non-articular depressions are also of variable form and are described as fossae, pits, depressions, grooves, furrows, fissures, notches, etc. These non-articular eminences and depressions serve to increase the extent of surface for the attachment of ligaments and muscles, and are usually well marked in proportion to the muscularity of the subject. A short perforation is called a foramen, a longer passage a canal. 1. Development of the skeleton. The skeleton. The skeleton is of mesodermal origin, and may be divided into a. That of the trunk, axial skeleton, comprising the vertebral column, skull, ribs, and sternum, and b. That of the limbs, appendicular skeleton. The vertebral column. The notochord is a temporary structure and forms a central axis, around which the segments of the vertebral column are developed. Note 11. In the Amphioxus the notochord persists and forms the only representative of a skeleton in that animal. End of note. It is derived from the entoderm and consists of a rod of cells which lies on the ventral aspect of the neural tube and reaches from the anterior end of the midbrain to the extremity of the tail. On either side of it is a column of paraxial mesoderm which becomes subdivided into a number of more or less cubical segments, the primitive segments. These are separated from one another by intersegmental septa, and are arranged symmetrically on either side of the neural tube and notochord. To every segment a spinal nerve is distributed. At first each segment contains a central cavity, the myocoil, but this is soon filled with a core of angular and spindle-shaped cells. The cells of the segment become differentiated into three groups, which form respectively the cutis plate or dermatome, the muscle plate or myotome, and the sclerotome. The cutis plate is placed on the lateral and dorsal aspect of the myocoil, and from it the true skin of the corresponding segment is derived. The muscle plate is situated on the medial side of the cutis plate and furnishes the muscles of the segment. The cells of the sclerotome are largely derived from those forming the core of the myocoil, and lie next to the notochord. Fusion of the individual sclerotomes in an antero-posterior direction soon takes place, and thus a continuous strand of cells, the sclerotogenous layer, is formed along the ventrolateral aspects of the neural tube. The cells of this layer proliferate rapidly, and extending medialward surround the notochord. At the same time they grow backward on the lateral aspects of the neural tube and eventually surround it, and thus the notochord and neural tube are enveloped by a continuous sheath of mesoderm, which is termed the membranous vertebral column. In this mesoderm the original segments are still distinguishable, but each is now differentiated into two portions, an anterior, consisting of loosely arranged cells, and a posterior, of more condensed tissue. Between the two portions the rudiment of the intervertebral fibrocartilage is laid down. Cells from the posterior mass grow into the intervals between the myotomes of the corresponding and succeeding segments, and extend both dorsally and ventrally. The dorsal extensions surround the neural tube and represent the future vertebral arch, while the ventral extend into the body wall as the costal processes. The hinder part of the posterior mass joins the anterior mass of the succeeding segment to form the vertebral body. Each vertebral body is therefore a composite of two segments, being formed from the posterior portion of one segment and the anterior part of that immediately behind it. The vertebral and costal arches are derivatives of the posterior part of the segment in front of the intersegmental septum with which they are associated. This stage is succeeded by that of the cartilaginous vertebral column. In the fourth week two cartilaginous centres make their appearance, one on either side of the notochord. These extend around the notochord and form the body of the cartilaginous vertebra. A second pair of cartilaginous foci appear in the lateral parts of the vertebral bow, and grow backward on either side of the neural tube to form the cartilaginous vertebral arch, and a separate cartilaginous centre appears for each costal process. By the 8th week the cartilaginous arch has fused with the body, and in the 4th month the two halves of the arch are joined on the dorsal aspect of the neural tube. The spinous process is developed from the junction of the two halves of the vertebral arch. The transverse process grows out from the vertebral arch behind the costal process. In the upper cervical vertebrae a band of mesodermal tissue connects the ends of the vertebral arches across the ventral surfaces of the intervertebral fibrocartilages. This is termed the hypocaudal bar or brace. In all except the first it is transitory and disappears by fusing with the fibrocartilages. In the atlas, however, the entire bow persists and undergoes chondrification. It develops into the anterior arch of the bone, while the cartilage representing the body of the atlas forms the dens or odontoid process which fuses with the body of the second cervical vertebra. The portions of the notochord which are surrounded by the bodies of the vertebrae atrophy and ultimately disappear, while those which lie in the centres of the intervertebral fibrocartilages undergo enlargement, and persist throughout life as the central nucleus pulposus of the fibrocartilages. The ribs. The ribs are formed from the ventral or costal processes of the primitive vertebral bows, the process extending between the muscle plates. In the thoracic region of the vertebral column the costal processes grow lateralward to form a series of arches, the primitive costal arches. As already described, the transverse process grows out behind the vertebral end of each arch. It is at first connected to the costal process by continuous mesoderm, but this becomes differentiated later to form the costo-transverse ligament. Between the costal process and the tip of the transverse process the costo-transverse joint is formed by absorption. The costal process becomes separated from the vertebral bow by the development of the costocentral joint. In the cervical vertebrae, the transverse process forms the posterior boundary of the foramen transversarium, while the costal process corresponding to the head and neck of the rib fuses with the body of the vertebra, and forms the anterolateral boundary of the foramen. The distal portions of the primitive costal arches remain undeveloped. Occasionally the arch of the seventh cervical vertebra undergoes greater development, and by the formation of costovertebral joints is separated off as a rib. In the lumbar region the distal portions of the primitive costal arches fail. the proximal portions fuse with the transverse processes to form the transverse processes of descriptive anatomy. Occasionally a movable rib is developed in connection with the first lumbar vertebra. In the sacral region, costal processes are developed only in connection with the upper three, or it may be four, vertebrae. The processes of adjacent segments fuse with one another to form the lateral part of the sacrum. The coccygeal vertebrae are devoid of costal processes. The sternum. The ventral ends of the ribs become united to one another by a longitudinal bar termed the sternal plate, and opposite the first 7 pairs of ribs these sternal plates fuse in the middle line to form the manubrium and body of the sternum. The xiphoid process is formed by a backward extension of the sternal plates. The skull. Up to a certain age the development of the skull corresponds with that of the vertebral column, but it is modified later in association with the expansion of the brain vesicles, the formation of the organs of smell, sight and hearing, and the development of the mouth and pharynx. The notochord extends as far forward as the anterior end of the midbrain, and becomes partly surrounded by mesoderm. The posterior part of this mesodermal investment corresponds with the basilar part of the occipital bone, and shows a subdivision into 4 segments, which are separated by the roots of the hypoglossal nerve. The mesoderm then extends over the brain vesicles, and thus the entire brain is enclosed by a mesodermal investment, which is termed the membraneous cranium. From the inner layer of this the bones of the skull and the membranes of the brain are developed. From the outer layer the muscles, blood vessels, true skin, and subcutaneous tissues of the scalp. In the shark and dogfish this membranous cranium undergoes complete chondrification, and forms the cartilaginous skull or chondrocranium of these animals. In mammals, on the other hand, the process of chondrification is limited to the base of the skull, the roof and sides being covered in by membrane. Thus the bones of the base of the skull are preceded by cartilage, those of the roof and sides by membrane. The posterior part of the base of the skull is developed around the notochord and exhibits a segmented condition analogous to that of the vertebral column, while the anterior part arises in front of the notochord and shows no regular segmentation. The base of the skull may therefore be divided into a caudal or vertebral, and b a precaudal or prevertebral portion. In the lower vertebrates 2 pairs of cartilages are developed, viz., a pair of paracaudal cartilages, one on either side of the notochord, and a pair of precaudal cartilages, the trabeculi cranii, in front of the notochord. The paracaudal cartilages unite to form a basilar plate, from which the cartilaginous part of the occipital bone and the basa sphenoid are developed. On the lateral aspects of the paracaudal cartilages the auditory vesicles are situated, and the mesoderm enclosing them is soon converted into cartilage, forming the cartilaginous ear capsules. These cartilaginous ear capsules, which are of an oval shape, fuse with the sides of the basilar plate, and from them arise the petrous and mastoid portions of the temporal bones. The trabeculi cranii are 2 curved bars of cartilage which embrace the hypophysis cerebri. Their posterior ends soon unite with the basilar plate, while their anterior ends join to form the ethmoidal plate, which extends forwards between the forebrain and the olfactory pits. Later the trabeculi meet and fuse below the hypophysis, forming the floor of the fossa hypophyseos and so cutting off the anterior lobe of the hypophysis from the spermodeum. The median part of the ethmoidal plate forms the bony and cartilaginous parts of the nasal septum. From the lateral margins of the trabeculi cranii 3 processes grow out on either side. The anterior forms the ethmoidal labyrinth, and the lateral and alar cartilages of the nose. The middle gives rise to the small wing of the sphenoid, while from the posterior the great wing and lateral pterygoid plate of the sphenoid are developed. The bones of the vault are of membranous formation, and are termed dermal or covering bones. They are partly developed from the mesoderm of the membranous cranium, and partly from that which lies outside the entoderm of the foregut. They comprise the upper part of the occipital squama, interparietal, the squamae and tympanic parts of the temporals, the parietals, the frontal, the vomer, the medial pterygoid plates, and the bones of the face. Some of them remain distinct throughout life, e.g. parietal and frontal, while others join with the bones of the chondrocranium, e.g. interparietal, scremi of the temporals, and medial pterygoid plates. Recent observations have shown that, in mammals, the basicranial cartilage, both in the caudal and precaudal regions of the base of the skull, is developed as a single plate which extends from behind forward. In man, however, its posterior part shows an indication of being developed from two chondrifying centres which fuse rapidly in front and below. The anterior and posterior thirds of the cartilage surround the notochord, but its middle third lies on the dorsal aspect of the notochord, which in this region is placed between the cartilage and the wall of the pharynx. End of section 9. Section 10 of Gray's Anatomy, Part 1. Anatomy of the Human Body, Part 1, by Henry Gray. Bone, Part 1. Structure and physical properties. Bone is one of the hardest structures of the animal body. It possesses also a certain degree of toughness and elasticity. Its colour, in a fresh state, is pinkish-white externally and deep red within. On examining a section of any bone, it is seen to be composed of two kinds of tissue, one of which is dense in texture, like ivory, and is termed compact tissue. The other consists of slender fibres and lamellae, which join to form a reticular structure. This, from its resemblance to lattice work, is called cancellous tissue. The compact tissue is always placed on the exterior of the bone, the cancellous in the interior. The relative quality of these two kinds of tissue varies in different bones, and in different parts of the same bone, according as strength or lightness is requisite. Close examination of the compact tissue shows it to be extremely porous, so that the difference in structure between it and the cancellous tissue depends merely upon the different amounts of solid matter, and the size and number of spaces in each. The cavities are small in the compact tissue and the solid matter between them abundant, while in cancellous tissue the spaces are large and the solid matter is in smaller quantity. Bone during life is permeated by vessels, and is enclosed, except where it is coated with articular cartilage, in a fibrous membrane, the periosteum, by means of which many of these vessels reach the hard tissue. If the periosteum be stripped from the surface of the living bone, small bleeding points are seen which mark the entrance of the periosteal vessels, and on section during life every part of the bone exudes blood from the minute vessels which ramify in it. The interior of each of the long bones of the limbs presents a cylindrical cavity filled with marrow and lined by a highly vascular areolar structure, called the medullary membrane. The strength of bone compared with other materials. Substance: medium steel. Weight in pounds per cubic foot, 490. Ultimate strength pounds per square inch. Tension 65,000. Compression 60,000. Shear 40,000. Substance: granite. Weight in pounds per cubic foot 170. Ultimate strength pounds per square inch. Tension 1,500. Compression 15,000. Shear 2,000. Substance: oak, white. Weight in pounds per cubic foot 46. Ultimate strength pounds per square inch. Tension 12,500. Footnote: indicates stresses with the grain, i.e., when the load is parallel to the long axis of the material, or parallel to the direction of the fibres of the material. End footnote. Compression 7,000. Shear 4,000. Footnote: indicates unit stresses across the grain, i.e., at right angles to the direction of the fibres of the material. End footnote. Substance: compact bone, low. Weight in pounds per cubic foot 119. Ultimate strength pounds per square inch. Tension 13,200. Compression 18,000. Shear 11,800. Substance: compact bone, high. Weight in pounds per cubic foot. Left blank. Ultimate strength pounds per square inch. Tension 17,700. Compression 24,000. Shear 7,150. Periosteum. The periosteum adheres to the surface of each of the bones in nearly every part, but not to cartilaginous extremities. When strong tendons or ligaments are attached to a bone, the periosteum is incorporated with them. It consists of two layers closely united together, the outer one formed chiefly of connective tissue, containing occasionally a few fat cells, the inner one of elastic fibres of the finer kind, forming dense membranous networks, which again can be separated into several layers. In young bones the periosteum is thick and very vascular, and is intimately connected at either end of the bone with the epiphyseal cartilage, but less closely with the body of the bone, from which it is separated by a layer of soft tissue, containing a number of granular corpuscles or osteoblasts, by which ossification proceeds on the exterior of the young bone. Later in life the periosteum is thinner and less vascular, and the osteoblasts are converted into an epithelioid layer on the deep surface of the periosteum. The periosteum serves as a nidus for the ramification of the vessels previous to their distribution in the bone, hence the liability of bone to exfoliation or necrosis when denuded of this membrane by injury or disease. Fine nerves and lymphatics, which generally accompany the arteries, may also be demonstrated in the periosteum. Marrow. The marrow not only fills up the cylindrical cavities in the bodies of the long bones, but also occupies the spaces of the cancellous tissue and extends into the larger bony canals, Haversian canals, which contain the blood vessels. It differs in composition in different bones. In the bodies of the long bones the marrow is of a yellow colour and contains, in 100 parts, 96 of fat, 1 of areolar tissue and vessels, and 3 of fluid with extractive matter. It consists of a basis of connective tissue supporting numerous blood vessels and cells, most of which are fat cells, but some are marrow cells, such as occur in the red marrow to be immediately described. In the flat and short bones, the articular ends of the long bones, in the bodies of the vertebrae, in the cranial diploe, and in the sternum and ribs of the marrow is of a red colour, and contains in 100 parts, 75 of water, and 25 of solid matter consisting of cell globulin, nucleoprotein, extractives, salts, and only a small proportion of fat. The red marrow consists of a small quantity of connective tissue, blood vessels, and numerous cells, some few of which are fat cells, but the great majority are roundish nucleated cells, the true marrow cells of Colica. These marrow cells proper, or myelocytes, resemble in appearance lymphoid corpuscles, and like them are amoeboid. They generally have a hyaline protoplasm, though some show granules either oxyphil or basophil in reaction. A number of eosinophil cells are also present. Among the marrow cells may be seen smaller cells, which possess a slightly pinkish hue. These are the erythroblasts or normoblasts, from which the red corpuscles of the adult are derived, and which may be regarded as descendants of the nucleated coloured corpuscles of the embryo. Giant cells, myeloplaxes, osteoclasts, large, multinucleated protoplasmic masses are also to be found in both sorts of adult marrow, but more particularly in red marrow. They were believed by Colica to be concerned in the absorption of bone matrix, and hence the name which he gave to them, osteoclasts. They excavate in the bone small shallow pits or cavities, which are named Howship's foveolae, and in these they are found lying. Vessels and nerves of bone. The blood vessels of bone are very numerous. Those of the compact tissue are derived from a close and dense network of vessels ramifying in the periosteum. From this membrane vessels pass into the minute orifices in the compact tissue, and run through the canals which traverse its substance. The cancellous tissue is supplied in a similar way, but by less numerous and larger vessels, which, perforating the outer compact tissue, are distributed to the cavities of the spongy portion of the bone. In the long bones, numerous apertures may be seen at the ends near the articular surfaces. Some of these give passage to the arteries of the larger set of vessels referred to, but the most numerous and largest apertures are for some of the veins of the cancellous tissue, which emerge apart from the arteries. The marrow in the body of a long bone is supplied by one large artery, or sometimes more, which enters the bone at the nutrient foramen, situated in most cases near the centre of the body, and perforates obliquely the compact structure. The medullary or nutrient artery, usually accompanied by one or two veins, sends branches upward and downward, which ramify in the medullary membrane and give twigs to the adjoining canals. The ramifications of this vessel anastomose with the arteries of the cancellous and compact tissues. In most of the flat, and in many of the short spongy bones, one or more large apertures are observed, which transmit to the central parts of the bone vessels corresponding to the nutrient arteries and veins. The veins emerge from the long bones in three places. Colica. 1. One or two large veins accompany the artery. 2. Numerous large and small veins emerge at the articular extremities. 3. Many small veins pass out of the compact substance. In the flat cranial bones the veins are large, very numerous, and run in tortuous canals in the diploic tissue, the sides of the canals being formed by thin lamellae of bone, perforated here and there for the passage of branches from the adjacent cancelli. The same condition is also found in all cancellous tissue, the veins being enclosed and supported by osseous material and having exceedingly thin coats. When a bone is divided, the vessels remain patulous, and do not contract in the canals in which they are contained. Lymphatic vessels, in addition to those found in the periosteum, have been traced by Cruikshank into the substance of bone, and Klein describes them as running in the Haversian canals. Nerves are distributed freely to the periosteum, and accompany the nutrient arteries into the interior of the bone. They are said by Colica to be most numerous in the articular extremities of the long bones, in the vertebrae, and in the larger flat bones. Minute anatomy. A transverse section of dense bone may be cut with a saw and ground down until it is sufficiently thin. If this be examined with a rather low power, the bone will be seen to be mapped out into a number of circular districts, each consisting of a central hole surrounded by a number of concentric rings. These districts are termed Haversian systems. The central hole is an Haversian canal, and the rings are layers of bony tissue arranged concentrically around the central canal and termed lamellae. Moreover, on closer examination it will be found that between these lamellae, and therefore also arranged concentrically around the central canal, are a number of little dark spots, the lacunae, and that these lacunae are connected with each other and with the central Haversian canal by a number of fine dark lines, which radiate like the spokes of a wheel and are called canaliculi. Filling in the irregular intervals which are left between these circular systems are other lamellae, with their lacunae and canaliculi running in various directions, but more or less curved. They are termed interstitial lamellae. Again, other lamellae, found on the surface of the bone, are arranged parallel to its circumference. They are termed circumferential, or by some authors primary or fundamental lamellae, to distinguish them from those laid down around the axis of the Haversian canals, which are then termed secondary, or special lamellae. The Haversian canals, seen in a transverse section of bone as round holes at or about the centre of each Haversian system, may be demonstrated to be true canals if a longitudinal section be made. It will then be seen that the canals run parallel with the longitudinal axis of the bone for a short distance and then branch and communicate. They vary considerably in size, some being as much as 0.12 mm in diameter. The average size is, however, about 0.05 mm. Near the medullary cavity the canals are larger than those near the surface of the bone. Each canal contains one or two blood vessels, with a small quantity of delicate connective tissue and some nerve filaments. In the larger ones there are also lymphatic vessels, and cells with branching processes which communicate, through the canaliculi, with the branched processes of certain bone cells in the substance of the bone. Those canals near the surface of the bone open upon it by minute orifices, and those near the medullary cavity open in the same way into this space, so that the whole of the bone is permeated by a system of blood vessels running through the bony canals in the centres of the Haversian systems. The lamellae are thin plates of bony tissue encircling the central canal, and may be compared, for the sake of illustration, to a number of sheets of paper pasted one over another around a central hollow cylinder. After macerating a piece of bone in dilute mineral acid, these lamellae may be stripped off in a longitudinal direction as thin films. If one of these be examined with a high power of the microscope, it will be found to be composed of a finely reticular structure, made up of very slender transparent fibres, decussating obliquely, and coalescing at the points of intersection. These fibres are composed of fine fibrils identical with those of white connective tissue. The intercellular matrix between the fibres is impregnated by calcareous deposit which the acid dissolves. In many places the various lamellae may be seen to be held together by tapering fibres, which run obliquely through them, pinning or bolting them together. They were first described by Sharpe, and were named by him perforating fibres. The lacunae are situated between the lamellae, and consist of a number of oblong spaces. In an ordinary microscopic section, viewed by transmitted light, they appear as fusiform opaque spots. Each lacuna is occupied during life by a branched cell, termed a bone cell or bone corpuscle, the processes from which extend into the canaliculi. The canaliculi are exceedingly minute channels, crossing the lamellae and connecting the lacunae with neighbouring lacunae and also with the Haversian canal. From the Haversian canal a number of canaliculi are given off, which radiate from it and open into the first set of lacunae between the first and second lamellae. From these lacunae a second set of canaliculi is given off. These run outward to the next series of lacunae, and so on until the periphery of the Haversian system is reached. Here the canaliculi given off from the last series of lacunae do not communicate with the lacunae of neighbouring Haversian systems, but after passing outward for a short distance form loops and return to their own lacunae. Thus every part of an Haversian system is supplied with nutrient fluids derived from the vessel in the Haversian canal and distributed through the canaliculi and lacunae. The bone cells are contained in the lacunae, which however they do not completely fill. They are flattened and nucleated branched cells, homologous with those of connected tissue. The branches, especially in young bones, pass into the canaliculi from the lacunae. In thin plates of bone, as in the walls of the spaces of cancellous tissue, the Haversian canals are absent, and the canaliculi open into the spaces of the cancellous tissue, medullary spaces, which thus have the same function as the Haversian canals. End of section 10.